Good morning to all. So, yesterday we completed, that is about the protein synthesis and the first half transcription. So, there was the DNA, so which was presented the genetic information, hereditary information inside the DNA. So, that was the DNA converted to so a Xerox copy of the mRNA converted to a Xerox copy of DNA. That is the information got transmitted from the DNA to mRNA. So, that phenomena was considered as a transcription. So, those are transcription phenomena. So, mainly the transcription was mainly occurred inside the, so that was takes place inside the nucleus. So, from the DNA to mRNA. So, that was mainly initiation, elongation and termination of the three processes are there. So, that was mainly the information was completely got transmitted from the DNA to mRNA. So, that was we are completed in a previous class. Today we are going to discuss about the, so that was a complete information about the protein synthesis. After completion of transcription, the translation is there. So, today is our topic, that is a translation. So, first DNA to mRNA, mRNA to tRNA. So, the transcription and translation. So, from the information was transmitted DNA to mRNA, that is a transcription. So, yesterday we complete about the transcription process, and mRNA to tRNA, so that is a translation process, that is the second process. So, that is a translation is mRNA to tRNA. So, how the translation process was occurred. So, today is our topic, we are going to discuss about the complete information about the translation now. So, mainly the ribosomes, the genetic information from the nucleus of the cell. So, it was mainly from the nucleus to the cytoplasm of the cell. So, the ribosomes, it was attached on the surface of the ribosomes. The ribosomes are mainly, so that was 70S ribosomes inside the prokaryotic cells, and the 80S ribosomes in eukaryotic cells. So, the 30S and 50S are in the subunits, and 60S plus 40S subunits inside the 80S eukaryotic cells. So, these are the small and the large subunits of ribosomes. So, these can play a major role in protein synthesis end process, that is a translation process. Why? Because, so before going to commencement of translation process, the information now is in the form of mRNA, that is in codons in the form of, that was a the information was in the form of genetic codons. So, those codons information was got transmitted from the mRNA to tRNA. So, before going to translation process, the ribosome two subunits are there. Those two subunits of ribosomes are going to become, so these are going to be attached with so one of another. So, then only those two subunits are first so combined with each other, so they can form a complex like structure, then only the translation process was started. So it is the ribosome structure. So the ribosome, the mRNA was so mRNA strand was going to be, so on the surface of mRNA strand, the ribosome two subunits are going to be attached with so two subunits are going to be form a complex like structure. So those complex like structure was formed. Then what happens is the genetic information which was presented inside on the surface of the mRNA, that was in the form of codons. Those codons are going to bonded with the anticodons which are presented on the surface of tRNA. So both are combined with each other, the codons are bonded with the anticodons, then so it can form a amino acid chain. So amino acids are formed fulfilled with the help of these. So it is a methionine, AUG is a methionine. So it was the genetic code. So it was going to indicate so one of the amino acid, that is a methionine. So these is always the initiator. It is always the initiator. The methionine was the initiator now. So it was going to start the translation process. So when the information got transmitted from the translated mRNA to tRNA, so the tRNA was going to attached on the surface of the ribosomes. So before going to attachment of the tRNA, the ribosomes two units are combined with each other. So those two units are combined, then the tRNA was going to attach, and so it is a E site, P site and the A site. So it was a landing site and it is a exit site, exit and a landing site. Means so the amino acids are going to be attached on the surface of the tRNA of a ribosomal larger unit, subunit. So first of all they are going to attach with the A subunit. So that was it was the A site, landing site, the fresh code, and amino acid, that is the codons are going to attach on the surface of first this A site, and it was going to transfer it to the P site. So then only the amino acid chain was going to be, so that was going to be continued and prolonged, that was going to be continued, that was a continuous process was occurred. So then the exit process. So after conversion of amino acids, those are the exit unit through this exit site, those are adjusted out from the external side. So the amino acids are free now. So those are bonded with each other. Before going to start this process, amino acyl tRNA synthesis. Amino acyl tRNA synthetase enzyme. So these enzyme was first going to be, so it was going to activate the tRNA. So amino acyl tRNA synthetase was one of the enzyme. First of all, these enzyme was going to be, so activates the tRNA, then only the tRNA was going to attach on the surface of the ribosomal larger subunit. So then the amino acid, that was the mRNA information in the form of codons, are going to be combined with the anticodons which are presented inside the tRNA. So it is a methionine and it is the isoleucine, and this is the serine amino acid, and UAA was the stop codon. So mainly, So AUG was always it was going to convert that was a methionine. Methionine is always the initiator codon. So it was the first amino acid. It was going to be attached on the surface of the tRNA of a ribosomal larger subunit. Then only the translation process was started. So it was the initiator. So then the another so one by one amino acids are going to be bonded with each other. So after the methionine, the isoleucine is another amino acid, serine is another amino acid, and the UAA was a, so do not code any amino acid. So it was also considered as a nonsense codon. Nonsense codon. So and it was helpful for the end codon or the stop codon. So the amino acid chain was continued. So that was a at the time of continuation of amino acid chain, when so that was codons are reached UAG, UGA or UGA. So those three codons are nonsense codons. Those codons may not be going to be coded any genetic amino acid. So these three codons are reached the amino acid chain, then immediately the amino acid chain was so the continuation of translation process was stopped at there. So that is the end process. So that was a completely end process was occurred when these stop codons are reached at there. Immediately the chain was going to be stopped at there. It is a smaller subunit of ribosome So see here now is only the small subunit of ribosome is only presented, but there is no larger subunit of ribosome. So before going to attach with the larger subunit, so that was the AUG was a initiator codon which was presented inside the mRNA, that was going to bond with the anticodons of tRNA, that is a methionine, and a smaller subunit of so now the complex is formed. See here in the next step, so it is a larger subunit and it is a smaller subunit, so both are formed combined to form a complex like structure here. So it is a larger subunit and it is a smaller subunit, both are fused to form together form so a complex like structure, then only the tRNA was easily attached on the surface of it, and the initiator codon was first scanned. So these complex is going to scan the first codon, then starts the translation process. So it was going to be scans, means identified first of all. So those the first codon, initiator codon, was first scanned by the complex of ribosomes, means the complex is nothing but which was made by the combination of larger subunit and another, that is a smaller subunit. So both are fused to form together to form a complex like structure. Then after the formation of these complex, it was going to scans the first, that is the initiator codon. So those initiator codon was scanned, then only the translation process begins. So in these initiation, elongation and termination, so 3 process and 3 steps are involved in these process. See this. So in these manner, so another information, first the AUG was first the initiator and the remaining amino acids are and remaining codons are formed. So the continued, so it is the initiation first, the initiation process is the main here. The initiation is the first step, that was mainly the AUG, the initiator codon was so going to attach, that was going to combine with the anticodons which are presented on the surface of the tRNA. So those are bonded and form the methionine. So then the remaining codons are continued. So this was mainly the mRNA was going to be synthesized. The mRNA chain, the strand of mRNA was continued, so in a 5' to 3' end direction. So in this direction the chain was continued without any interruption. So this continuation process was called as elongation. The chain was completely continued up to the, so one of the amino acid, the codons are started to came from the mRNA. So those are each and every codon which is going to bonded with the anticodons which are presented on the surface of the tRNA, both are bonded with each other and finally forms the amino acid, and so one by one those amino acid chain was continued. So the phenomena was considered as a elongation process, and the termination is last step here. The termination is the end process. So those after the formation of amino acids, so those amino acids are bonded with each other. So those are peptidal transferase. The enzyme peptidyl transferase, so these peptidyl transferase was used to form so amino acid, see this. So it is a amino acid chain. In the middle of each and every amino acid, the bond is formed with the help of these peptidyl transferase was used to form the peptide bonds in the middle of amino acid. So amino acid chain is it was continued like this. So in this way, so that was terminated means ended. So that was with the help of end codons. When the end codons are reached to the on the surface of the tRNA, then so the translation process is completely stopped. So in these manner, see here, so it is the mRNA. So these are the codons, and the codons of mRNA are going to bonded with the anticodons which are presented on the surface of the tRNA. These two are the ribosomal small unit and the ribosomal larger subunits. These are, so these ribosomal large unit and subunits so what happens is bonded with each other in the middle of these mRNA presented. So mRNA, so on the surface of a larger subunit of ribosome tRNA is located. So see here, so these are the codons of mRNA are going to bonded with the anticodons of tRNA. See this, in this reaction the GTP was utilized here, the guanosine triphosphate was GDP, and phosphate was free phosphate was released, it was utilized in this reaction. So in these not only the AUG is the, so that was the first and initiator codon. Not only this, the remaining all are so that was another amino acids are along with so methionine, the serine, isoleucine. So those all are continued, and so the amino acid chain was the first see here on the surface of the mRNA. First, so it is the mRNA 5' to 3' in direction. These are the codons which are presented, and it is a larger subunit, and here the smaller subunit is there. So it is the E site and the A site and the P site. So freshly land, so amino acid was first going to be attached on the A site, that was transmitted to the P site, then only that was going to bonded with the codons of mRNA. So those are anticodons. First that was placed on the surface of the A site, that was transmitted to the P site, then the inside the P site, so those codons are bonded with the anticodons. Then so see here methionine, serine, isoleucine. So in this manner the amino acid chain was continued like this manner. So when the codons are bonded with the anticodons, first it was going to be placed on the surface of the A site, that was transmitted to the P site. After the conversion of bonding with the codons with the anticodons, so then that was completely fulfilled amino acid, that was again transmitted to the E site. It is a exit site. It is the exit site. From these exit site, so those free amino acids are so those are so released from the exit site. So those are combined with the one with the another. See this. The amino acids are continued, and so those are bonded with each other with the help of the enzyme peptidal transferase. Peptidal transferase is used to form the so peptide bonds in the middle of the two different amino acids. So in this way amino acid chain was continued till to the end of UAA, UGA, UAG. So these three are the nonsense codons or stop codons. So in these three codons the amino acid chain was that was raised to the from the methionine onwards to that was so received in these three codons or any amino acid. So then UAA or either UAA, UGA or UAG, then immediately the amino acid chain was going to be stopped. So then immediately that was what happens is the translation process ended, that is a termination process. At the end of termination, then immediately so in these three codons, any one of these three codons are going to be so attached on the surface of the exit A site and transmitted to P site. Then immediately the amino acid chain prolongation was stopped, the translation process is stopped, that is a termination process. Then immediately the free amino acid chain, it is a so number of the amino acids are fused to form polypeptide chain. So those polypeptide chain was completely got separated from the ribosomal larger subunit, that was reached to final their destination part. So then after, so this is these three processes which are involved inside the that was a translation. So it is today's topic. The today's topic is over. So we will meet tomorrow. Thank you.